Hello, good morning or good afternoon, everyone. I am Dr. Mohua Mukhopadhyay of Lady Brabourne College, Kolkata. Today, the topic of my discussion is endophytes of tea plants from Darjeeling district, West Bengal. So, Darjeeling is a very well-known hill station situated in West Bengal and obviously known as Queen of Hills. It is not only famous for its beautiful landscape, but also it is famous for tea plantation. It is processed as black, green, white, or oolong tea. So in the present study, I have chosen tea plant for the isolation of endophytes. So what are endophytes? Endophytes are microorganisms that live inside the living plant tissue for at least part of their life without causing any apparent disease symptoms in the host. They are treated as endosymbionts also. To present these types of microorganisms, D. Berry coined the term endophytes. So endophytes mainly entered inside the plants primarily through the roots or the aerial portion of the plants such as leaves, flowers, stems and cotyledons. They are localized at the point of their entry or they can spread in the whole host plant body. So after entering the host, they reside within the cells or within the intercellular spaces or in the vascular system, etc. So they are distributed in all over the host body. So after colonizing in the plant tissue, the endophytes are known to produce a diverse range of natural products which could be consistent and successful source of drugs and agricultural supplies. Therefore, the natural products from endophytic microorganisms do have a great potential role not only in the pharmaceutical industry but agrochemical and biotechnological industries also. So here we have chosen a very important and very well-known tea estate of Darjeeling, that is Happy Valley Tea Estate, and the tea plants are collected from that particular tea estate. They were taken to our laboratory and finally the endophytes are isolated. So what are the technique of isolation? They are mainly rigorous steps of surface sterilization is needed and to exclude the microorganism present outside the plant parts, and for that we have used the normal tap water, 20 sodium hypochlorite, 70% alcohol, and obviously the sterile distilled water. So after the complete surface sterilization, the slurry was prepared using saline water, sterile saline water, and they were plated in Luria Bartani agar and incubated at 37 degrees for 2 days. So after incubation, they produce certain colonies of bacteria as well as some fungal colonies are also produced. So in this method, we are able to isolate 14 bacterial and 6 fungal isolates. And they were studied in microscopic examination, and they are mostly either coccus or the bacillus in nature. And after that, whether these organisms are able to produce or able to produce the auxin and gibberellin. We know that these are two important growth promoters in plants. Whether these organisms are able to produce these auxin and gibberellin were studied in detail. And for gibberellic acid production or gibberellin production, we have used the method by Boro et al. And it is found that each and every strains of 14 bacterial strains, I can say, are potent producer of this gibberellic acid, and gibberellic acid ranges from 172 μg per ml to 383 μg per ml, so that we can say that these organisms are potent in gibberellic acid production and may help in plant growth in different respects. In case of auxin, we have estimated the auxin by Gordon et al. Method using Salkowski reagent, and all the isolates again are potent in producing the auxin in the appreciable level of 160 to 300 mg per ml quantity. So these bacterial isolates, I can say, are very potential in producing the growth promoters. And again, we have performed some experiment whether these organisms are able to solubilize phosphate or can fix atmospheric nitrogen, but these organisms don't show any phosphate solubilization or nitrogen fixation ability. Next, we pass on to the organisms whether they are having any antimicrobial activity or not. So we have chosen several bacteria, taste organisms, which are mainly pathogenic in nature, like Vibrio cholerae, Pseudomonas aeruginosa, Bartholdius epsia, then Acinetobacter baumannii and Klebsiella species, as well as the E. coli. And some of them are the test organisms, mainly the 3 test organisms, were found to show antibacterial activity against 3 pathogenic microorganisms out of the 6 test organisms. So we can say that they are having some broad spectrum antibiotic producer. They are belongs to that family, and that potential again may be exploited in the pharmaceutical industry. Next, we pass on to the fungal members. And it was found that the fungal members are potent in producing different types of enzymes, and they play a very important role in the elemental cycle. And, the 4 of the fungal isolates are can degrade proteases, can produce proteases, whereas all of the 6 fungal isolates are efficient in the degradation of starch by producing amylase. So this is all about the study. That is, we have elaborately studied the bacterial as well as the fungal isolates and their potential for the production of different types of biochemical products. So in conclusion, I can say that endophytes are very potent microorganisms which may be explored for the production of growth promoters, antimicrobials, and also different important exoenzymes. So thank you. I am just concluding my presentation. Thank you, the organizer, for giving me the platform to present such work.